Gentlemen, it's my pleasure to introduce Professor Christos Venetis, a well-known expert in reproductive medicine. He's an associate professor at the University of New South Wales, Australia, and the Aristotle University of Thessaloniki in Greece. And he's a well-known specialist in reproductive endocrinology, ovarian stimulation, and IVF outcomes. With a PhD and in reproductive endocrinology, extensive clinical experience, and expertise in big data research and epidemiology. Professor Venetis has authored over 95 publications and is ranked among the top 2% most cited researchers globally in his field. He's also in an editorial board of Human Reproduction Journal. Today, he will share his insights on follitropin alfa biosimilars, lessons from clinical trials, and real-world practice. Professor Venetis, floor is yours.
Thank you. Thank you, Professor Gryshchenko, for your kind introduction. And I would like to thank you and to thank Sona-Pharm for the invitation to be with my colleagues via this platform and to discuss the latest evidence on a topic which I think is of significant interest for all clinicians who on a daily basis have to deal with getting the best results from the cycles of their patients. And as we all know, choosing the right gonadotropin, choosing the right protocol and the right medication is crucial when someone endeavors in this task. So hopefully within the next half an hour, I'll be able to share with you the latest data on this topic as it pertains to the biosimilars of follitropin alfa compared to the reference product. And I hope that this will be a stimulating presentation and there's going to be an even more interesting discussion to follow. So I would like to start by sharing my slides. And I hope everyone can see that. So
Yes.
I will launch straight to the presentation. So these are my disclosures. And obviously, I think whenever someone starts looking into data, we always have to acknowledge the fact that reproductive medicine is essentially science. There is an element of art into it, that's for sure. But I think we all know that deep down in its core, medicine and therefore reproductive medicine is scientific. And when we refer to science, like the easiest probably definition I could find online is that science is the knowledge based on demonstrable and reproducible evidence. And you'll see that we'll come back to these two key concepts, the fact that it should be demonstrable and reproducible evidence. So the scientific method, as all of us know, starts by observing something interesting, observing something that sort of catches our eye, and then we form a hypothesis and we say, that's funny. Could it be that when we do this, that happens? So in order though to be able to prove that that was not just a chance observation, but this is something that truly happens in nature, we have to properly test the hypothesis. So we design an experiment, we standardize the experiment, and we test that hypothesis. And based on the results of that experiment, we can either accept or reject the hypothesis. And I think that is like the scientific method we all know. I would just like to add the final step that many of us seem to sort of forget, which is essentially we need to be able to reproduce the experiment and get the same results. And it is important to understand that if there is no reproducibility, what we're observing is probably not scientific and not close to the truth. So research aims to serve exactly this purpose, aims to help us understand which ones of the hypotheses we form are false, are incorrect, and which ones are true, and therefore form the basis of scientific knowledge. And here you can see the cycle of research. And it's interesting, for example, on many occasions, we have primary data, basic research that gives essentially life to a clinical hypothesis. And then based on the findings of basic research, we have what we call translational medicine, where we translate these basic research findings to some intervention for patients. And then obviously we have to test them. And depending on the results, we can then extend that into daily practice. And of course, the research does not end there. Essentially, when we're in daily practice, we constantly evaluate and try and make sure we confirm or not confirm what clinical trials have shown. So that's one way that we do research, and that's one way where we produce knowledge. But there are many other ways. I mean, as this is a circle, a cycle, you can actually enter the cycle wherever you want. So many times, clinicians will observe something in their daily practice, and they will form a hypothesis. And following that hypothesis, they can start testing that hypothesis by doing, for example, retrospective research, or by going back to basic research and trying to identify what is the pathophysiological basis of what they have observed. And by doing so, by confirming the truth via different methods of research, we know that what we're actually observing is certainly much closer to being true. And that's how in the 90s, the term evidence-based medicine was introduced, where essentially instead of each one of us relying on knowledge, textbooks, and personal experience, we started relying more and more on actual evidence that show whether something is likely to be true or not. This is the pyramid of evidence-based medicine that has been published by, Oxford Center for Evidence-Based Medicine. And you can see that expert opinions do not actually count that much. And what we have at the pinnacle of this pyramid, at the peak, is randomized controlled trials and obviously systematic reviews of randomized controlled trials. And just under these randomized controlled trials, we have cohort studies and case control studies and all other sources of evidence. So after this brief introduction on science and how we generate evidence and what's potentially the value or the relative value of this evidence, I would like just to go back closer to what this talk is about. Obviously, when we stimulate the ovaries, our aim is to collect multiple oocytes. And we want ideally to have good quality oocytes and to have a good number of them. What constitutes a good number of them probably is not the topic of today's discussion, but most people agree that it should be essentially somewhere around 10 to 15 or 15 to 20, depending on which study you look at. So the current paradigm of IVF involves us giving medication to women to induce multifollicular growth. And what medication do we give? We give FSH injections. So that specific medication seems to be very crucial in the whole paradigm of ovarian stimulation. And of course, by inducing multifollicular growth, we hope to retrieve multiple eggs, multiple oocytes, create multiple embryos, transfer usually the best one or two of them, depending on the age of the woman and what is allowed in each one's jurisdiction. And then if we have supernumerary good quality embryos, then we can freeze them so that that couple or that individual can use them in the future. So ovarian stimulation today is almost exclusively performed with gonadotrophins. And the history of gonadotrophins goes way back. They were essentially discovered in the 30s. And then probably the first gonadotrophin that was extracted by the human pituitary was in the late 60s. And following essentially the rapid popularization of assisted reproduction, either with ovulation induction in the beginning and IVF to follow that, we had essentially an explosion of discovery and biomechanics in terms of the development of new pharmaceutical products, including the highly purified FSH. And then in the mid-90s, we had the recombinant gonadotrophins, the recombinant follitropin alpha, beta, delta nowadays, cord follitropin, which is a chimeric product, and so on. And somewhere in the 2010s, many companies started focusing on producing biosimilars of follitropin because the recombinant FSH, like follitropin alpha and beta, had been the most popular gonadotrophins used to that point. So what are biosimilars? Biosimilars are not generics. They are biological medicinal products that contain a version of the active substance of an already authorized original biological medicinal product, which we call the reference product or the originator. And it should be emphasized that biosimilars are not identical molecules. They are similar. They are not identical, and therefore may still differ in strength, in purity, contain different composition of isoforms and other various glycosylation profiles, and that might mean consequent alterations in clinical efficacy or safety. Usually, biosimilars are being approved and granted authorization in the European and other markets by creating a comprehensive sort of file where they can establish similarity in terms of quality characteristics, biological activity, safety, and efficacy. And that stems usually from comparability studies before they can be granted approval and can be marketed as biosimilars. Interestingly, and this is something again that has very, very important implications for what we're discussing, the EMA and the FDA mandate that in order for a company to prove similar biological activity, they just need to prove that their product has essentially reduces or leads to a non-inferior number of oocytes retrieved compared to the originator product. So these studies are not performed to test live birth rates or ongoing pregnancy rates. They just test the number of oocytes retrieved and whether they are non-inferior or not. But whenever we want to choose the right treatment, we always have to essentially judge how good the treatment is on the basis of 4 pillars of health technology assessment. These pillars are efficacy. Is the method effective? Safety. Is it safe? It could be effective, but might not be safe. Is it acceptable by patients? I mean, there are exceptional medications that have very severe side effects, or, the route of administration is not acceptable by patients. And of course, from a public health policy perspective, we always have to be mindful of the cost. So the other big question is cost effective, whether they're cost effective. And the reason why we discuss that is because many biosimilars for many years, understandably so, IVF is a very expensive process for most individuals, have been budgeted and marketed as a more affordable and equally efficacious option for our patients. So we were very interested in looking into all these aspects when it comes to the biosimilars. And for that reason, we designed a systematic review and meta-analysis, which is actually an update of an earlier work. And the main question of this systematic review and meta-analysis was whether, based on the best available evidence, which obviously is randomized controlled trials, biosimilars of follitropin alfa are comparable to the originator in terms of live birth rates. So to answer this question, we went to the pinnacle of the hierarchy of evidence according to the pyramid of evidence by the Oxford Evidence-Based Medicine Center. And we designed a systematic review and meta-analysis according to the highest methodological standards, according to PRISMA guidelines. We pre-registered the protocol in the International Prospective Registry for systematic reviews. And we performed literature searches across many different electronic databases and clinical trial registries up until January 2024. Of course, there were no language limitations. We included only human studies, as you would imagine, and all duplicates were removed. And we also, according to and based on the recommendation made by the Cochrane collaboration, we also judged the quality of the available studies using the risk of bias 2 tool. As we discussed, the main outcome measure was live birth rate per randomized patient. And some people will say, and why is that, Dr. Venetis? Well, because patients come to us not necessarily to collect eggs, but collecting eggs is the means to an end. Patients come to us because they want to have a baby. And it has been, for some years now, probably close to 2 decades, agreed amongst reproductive medicine specialists that the most clinically relevant outcome for patients is and should be live birth rate. So live birth rate per intention to treat analysis, meaning per randomized patient, which is the methodologically more robust way to go, was our primary outcome measure. Of course, we also assessed ongoing pregnancy rate at 10 to 12 weeks of gestation and clinical pregnancy rate at 5 to 8 weeks of gestation. We also looked at multiples. We also tried to look at cumulative clinical pregnancy, the total dose of gonadotropin, the duration of ovarian stimulation, the number of oocytes retrieved per aspirated cycle, the number of good embryos, and of course, OHSS and other outcomes like miscarriage rate, ectopic, multiple pregnancy rate, as discussed, and so on. We wanted to be able to provide as much evidence regarding our assessment as possible. We also decided to perform a sensitivity analysis by excluding all the studies that would have scored as being at high risk of bias. And this is again according to the guidelines by the Cochrane collaboration. And we performed subgroup analysis according to the different types of biosimilars, to biosimilars by approved by the FDA and EMA versus biosimilars not going through the same rigorous testing, and also according to the type of patients. Are, for example, certain biosimilars, do they perform better compared to compared to others or compared to the originator, depending on the type of patients you have, whether you have high responders, normal responders, or poor responders, or at least that was the intention and that was a goal. As I said, all these were protocolized and were registered before we actually had a look at the evidence. So this is the PRISMA flowchart. And you can see that by going a very stepwise, meticulous process by 2 independent screeners, we ended up with 8 randomized controlled trials, including close to 3,000 randomized patients. These are the trials, and you can see you can see the number of registration because all these trials needed to be registered. That wasn't obviously something that it was an inclusion criteria for our systematic review and meta-analysis, but given that all these trials were used for regulatory purposes for these biosimilars to obtain approval, they had to be registered. So we include both the registration number, but also the actual name of the biosimilar follitropin alfa that was tested in the study. You can see the sample size ranged anywhere from 100 to 1,100 approximately. All of them were randomized based on some type of computer-aided algorithm, and most of them were single-blinded, and there was one triple-blinded study. And you can see here that with the exception of one, all others had as a primary outcome measure the number of oocytes retrieved, because as we've discussed, they needed to prove non-inferiority in the number of oocytes retrieved. And there was one study that used clinical pregnancy and another one that used the percentage of M2 oocytes. On the basis of what other clinical protocols were employed, you can see that we have a mix of both agonist and antagonist cycles. And in regards to the doses, the starting doses that were used, anywhere between 150 to 225 for most studies. And in one study, there was also a different stratification, and the starting dose was essentially determined by the age of the woman. If she was less than 30, she would get 150 units per day. If she was 30 to 34, 150 to 225. And if she was 35 or older, she would be getting 225 and 300. All of them allowed modifications of the starting dose of FSH. And in terms of the studies, most of them included just one cycle. And there were some studies that included more than one cycle. However, just to keep things fair and methodologically robust, we included data in this meta-analysis only from the first cycle so that we can have the most, as I said, methodologically robust result. This is the quality assessment as performed by 2 independent reviewers using the ROB2 tool. And you can see that generally the studies are of high quality. However, we had 3 studies that were found to be at high risk of bias, and usually main to deviations from intended intervention, and in some cases due to missing outcome data. And these are relevant information for you because, as I mentioned earlier, these studies were excluded in a sensitivity analysis we subsequently performed. So let's start with a big question. Is there any difference in the live birth per randomized patient between patients receiving biosimilars of follitropin alfa versus those who received the originator? And as you can see here, our meta-analysis suggests that yes, indeed, patients that have been stimulated with a biosimilar to follitropin alpha had about a 17% relative decrease in the chance of having a live birth compared to the originator. As you can see, the relative risk is 0.83, and it's statistically significant as the confidence interval does not cross 1. That means if we accept that 17% relative risk difference, that is something that we all need to sort of take into account whenever we're counseling patients about the right type of gonadotrophin. However, let's see whether this evidence is also confirmed by other outcomes. When we looked at ongoing pregnancy per randomized patient, you can see here that again, having been stimulated with biosimilars to follitropin alpha led to significantly lower ongoing pregnancy rate per randomized patient compared to being stimulated with the originator with a reference product. The relative risk is 0.81, so the reduction is about 19% in relative risk. And something that's very important, and I did not mention in the previous slide as well, you can see that heterogeneity is essentially absent. There's 0% heterogeneity. And with the exception of the Pasqualini study, which is the one with the widest confidence interval, it's almost as if all the other studies agree that the relative risk favors the originator. It is just that for all studies, potentially due to their low sample size, they were not able to prove that indeed there is a significant difference. And when they were combined using a meta-analysis, the overall power was increased, and all of a sudden now this effect size is statistically significant. Moving on to clinical pregnancy, again, very similar story. Statistically significant decrease of around 18% in clinical pregnancy rate per randomized patient for those who were stimulated with the biosimilar to follitropin alpha compared to those who were stimulated with the reference product with the originator. Again, heterogeneity almost completely absent. The study by Pasqualini is the only one who is not in the same direction of effect, but that's also the smallest one, and you can see how wide of a confidence interval it has. When we looked at secondary outcome measures, obviously we would start with the number of COCs retrieved, which, as we said, was a primary outcome for most of these studies. And you can see that there is no statistically significant difference. The weighted mean difference was plus 0.3 approximately oocytes in favor of the biosimilars. However, this was not statistically significant. When we looked at OHSS, relative risk 1.17, no significant difference. There was a slight difference in the duration of gonadotrophin administration. So the individuals who were stimulated with the biosimilars of follitropin alpha had about, on average, 0.2 days less of ovarian stimulation compared to those stimulated with the reference product. And there was no significant difference in the total FSH dose used. Finally, and importantly, when we did the sensitivity analysis by excluding all the studies, the 3 studies that we had found at high risk of bias, you can see here that once again, live birth, ongoing pregnancy, and clinical pregnancy were all statistically significantly inferior in patients stimulated with the biosimilars of follitropin alpha compared to those stimulated with the reference product. We also, as promised, performed subgroup analysis depending on the type of the biosimilar, whether they had received FDA or EMA approval or not, and whether they had a specific type of population. And unfortunately, we found only 2 types of population being included in the eligible studies, either normal responders or unclassified general population. And there was no indication that there is some type of significant difference between these subgroups. So, based on this meta-analysis, which has been very recently published in Human Reproduction, we can see that the latest randomized evidence suggests that live birth, ongoing pregnancy, and clinical pregnancy rates are significantly lower with biosimilars of follitropin alpha compared with the reference product. And this is despite the fact that the number of oocytes retrieved are very, very similar. There is no difference there. But these are RCTs, and there are just 8 RCTs. And yes, it's about 3,000 patients, but many people, for some reasons, might also want to see that the findings of the RCTs are also essentially confirmed by more real-world evidence. So when we do have that, when we see that the findings of the RCTs are also confirmed by real-world evidence, essentially we have what we call the triangulation of evidence, and that increases our confidence in the findings. So to shed some further light on that, I would like to briefly present to you the SNIRAM study. The SNIRAM study is a real-world evidence study coming from France, published by Michael Greenberg. And the target or the aim of this study was to describe the ART pathway and, importantly, to compare the effectiveness between different gonadotropins, both in terms of live birth and cumulative live birth rates. It essentially employed the use of all the French national database, including at least 99% of the population. And as you can see here, it included more than 150,000 patients. Looking at the cohorts that were constructed, you can see that there was one cohort of follitropin alfa originator, of other originators, of urinaries, and of follitropin alfa biosimilars. And obviously, we will be focusing on the comparison between the originator follitropin alfa and the biosimilars. As I said, more than 150,000 women were included, including 245,000 stimulations. And the mean age of the women were about 34, and about 10% of the population had performed freezal. So going straight to the results, if we can see here, the live birth rate, the odds were very, very similar to what we observed in terms of relative risk, was about 19% lower chance of live birth with biosimilars compared to with the originator. And this is an adjusted odds ratio where they have essentially adjusted for multiple confounders. The cumulative live birth rates, something that unfortunately we didn't have any data for in the randomized control trials, were also significantly lower at 0.86 with a confidence interval 0.82 to 0.89. So there was a statistically significant increase in about 14% in the odds of having cumulative live birth with biosimilars compared to the reference product. They also looked at the different age groups to see maybe the directional effect was changed in these age groups, and you can see that it remained pretty consistent across all age groups, and also on whether patient had done freezal or not. At the same time, they did another type of sensitivity analysis, which is called the propensity score matching, using only the first cycle of each patient. This is considered a very advanced statistical procedure, trying to essentially mitigate even more or eliminate, if possible, any residual confounding and produce the most unbiased estimates. Because as you can imagine, this is observational data. This is not randomized data. So these type of methodologies are required to get closer to the truth. And once again, even with a propensity score model, there was a reduction in the cumulative live birth with the biosimilars compared to the originator. You can see here that the hazard ratio was 0.87 from 0.81 to 0.93. So a significant reduction there as well. So by looking at real-world evidence as well, they seem to suggest that live birth and cumulative live birth rates are also significantly lower with biosimilars of follitropin alfa compared with a reference product. So the final section of this presentation is, why are these differences there? First of all, we already discussed that similar is not identical. And unfortunately, the studies that have been published were not able to give us some further insight on whether there were, for example, differences in the type of embryos that we were able to create. We can look at more basic data and see that, for example, a specific biosimilar follitropin alfa has a different profile of glycosylation with a higher proportion of isoforms with tri and tetracylated, and this means that it will have a difference in vivo bioactivity. And this difference in vivo bioactivity compared to differences into batch-to-batch coefficient of variation might be one of the explanations for the observed differences. There are other basic data as well, where they point to essentially different reactions in terms of the intracellular calcium ions production. And these differences might be there to support why we see these absolute clinical differences. And of course, another study by Manti and colleagues published in 2022 showed that there's actually, in terms of post-translational modifications between different biosimilars of follitropin alfa and the originator, it appeared that we had higher average oxidation for some of the biosimilars compared to the reference product. All these are indirect findings and could be circumstantial, but these clearly showcase that there are differences at the molecular level, which might or might not be responsible for the observed clinical differences. The truth of the matter is we don't really know the clinical differences, why the clinical differences are there. So we're just looking into basic research and try to identify whether there are specific hints that could potentially help us essentially reveal that pathway. So the differences between the biosimilars and the originator for the follitropin alpha, as I said, seem to exist. However, we do not currently fully understand whether these are responsible for the observed clinical differences. So finally, for the last 2 minutes, I know, as I said in the beginning of my talk, that many of us have been using biosimilars because they seem to represent an affordable and more potentially cost-effective alternative compared to the originator. And we know that cost is a big consideration in IVF. So in order to prove whether they are truly cost-effective, we performed a cost-effectiveness analysis using RCTs data. And you can see here that the incremental cost-effective ratio for the originator compared to the biosimilar was about 7,200 euros. That means that each extra live birth that we were getting was only costing 7,210 euros, which is considered pretty low. In most countries in Europe, we consider the threshold to be around 20,000 euros. Also, a study coming from France based on the Greenberg data, the data that I showcased you earlier, the real-world evidence, that was a different cost-effectiveness analysis, and it also confirmed that both live birth rate and cumulative live birth rate, the incremental cost-effectiveness ratio stayed way below 7,000 euros, 5,500 for live birth, and around 1,900 euros for cumulative live birth rate. So with both these cost effectiveness analyses, and I would suggest you go and read them yourselves, it's clearly shown that despite the fact potentially the originator in this instance being more expensive per unit, the fact that it's far more effective leads to essentially being the cost effective option compared to the biosimilars. So, dear colleagues, ladies and gentlemen, concluding my presentation, I hope it became clear that gonadotropins are an integral part of ovarian stimulation and that biosimilars of follitropin alpha have been introduced in the last decade, offering a more affordable alternative to the originator products. However, as with anything, we need to be very careful before using them and ensure that indeed they are safe, they're efficacious, they're acceptable by patients, and they're cost effective. The most recent meta-analysis performed, published just 2 months ago, suggests that actually biosimilars of follitropin alpha are associated with lower live birth rates, lower ongoing and clinical pregnancy rates compared to the originator, while leading to the same number of oocytes retrieved. Studies using real-world evidence seem to confirm these findings. We don't really know why these differences are there, but we have been able to see differences in the bioactivity and other characteristics of the biosimilars and the originator product of follitropin alpha. And furthermore, cost-effectiveness analyses have also shown that given those differences in efficacy, despite the fact that the originator usually being the most expensive product, it is still the most cost-effective product just because it leads to more live births. Of course, we should never be satisfied. These are all data we should be taking into account whenever counseling our patients and making our own decisions, but we should also urge everyone who's interested and everyone individually to perform and conduct further research in order to evaluate further the differences between the biosimilars and the originator follitropin alfa, and importantly, to identify what is the actual pathophysiological reason for the observed differences. And with that final message, I would like to thank you for your attention, and I'm looking forward to discussing these results with you. Thank you.
Thank you, Professor Venetis, for sharing this wonderful data, and especially thank you for this so elegant meta-analysis, this so robust methodology. And of course, we have questions from the auditorium, and let me ask you the first question. So the data from your latest meta-analysis showed that there's no difference in the number of oocytes when using biosimilars comparing to the originator. And that's the main difference from the previous one, from the previous meta-analysis, as I can see. But that's a very complicated field. But can I ask you to comment what could be the most likely explanation for a significant decrease in the live birth rate with use of biosimilars?
Thank you. Thank you, Professor Gryshchenko. And indeed, and I will be brutally honest with you and all my colleagues. When we started this exercise, and it started this exercise as part of essentially a master's project, even back in 2019, when we started the original project, we almost thought it was pointless and there would be no differences. But we said, look, we might as well do it. No one has looked into live birth rates, so we might as well just do it and confirm that indeed there is no difference. And we were quite surprised when the differences were there. We obviously try to identify what might be the reason. The truth of the matter is we still don't know. And the main reason we do not know is that the vast majority of the studies that have been performed have not actually looked into great detail in the embryological outcomes. They almost collected the data up until the number of oocytes retrieved, which was what was required by the regulatory authorities from them. And then they just didn't care about collecting more data, apart obviously from live birth rates and clinical pregnancy rates that they also had to do. So we were not able to see, at least clearly, whether there are differences, for example, in, let's say, the number of mature oocytes, the fertilization rates, the cleavage rates, the quality of the embryos produced. It should also be noted that we have absolutely no other evidence potentially, helping us understand whether the endometrial receptivity is affected, because all the data I showed you actually, pertain to the fresh embryo transfer cycle. In some cases, we had a couple of studies where, for reasons that were not clearly described in the manuscript, they had significantly more fresh embryo transfers with the originator compared to with the biosimilars, but they do not mention anywhere in their study why that was. So we need to look closer. And we need to, and I'll be honest, I personally feel that this is a very exciting field of research, because biosimilar products have been and will continue to be in many fields of medicine very, very important to essentially offer good quality products, affordable products to our patients. But we need to ensure that they are indeed as efficacious as the originator. And I feel that in this regard, the biosimilars of follitropin alfa might have missed the mark. I would like, and I would urge all the commercial entities that are essentially involved in the production of biosimilars to go back, look at how they produce their molecules, and try and identify where these differences might come from, and hopefully optimize the production of the biosimilars so we can have comparable outcomes. But currently, I'm afraid we are in a very clinically weird situation where we have randomized evidence and real world evidence that live birth rates are not the same, but at the same time, we have, biosimilars that have been approved as being biosimilar on the basis of just equivalence in terms of number of oocytes retrieved. So we need somehow to find answers. And for as long as we're not finding answers, I think that our confidence in the use of biosimilars will be essentially going down. I know that has happened, Professor Gryshchenko, for myself. I was using biosimilars quite regularly just because I felt it was an affordable way, especially for patients that couldn't afford the more expensive medication. But ever since this meta-analysis came out, I feel that it is, essentially something that the patients at least need to know. And most of the patients, when they look at the evidence, will choose not to go ahead with the biosimilars and pay a slight premium to buy a medication that leads to higher pregnancy rates because they know they are getting more back for their money's worth in a way.
Thank you so much. And based on your statement, how do you think the cost benefits of biosimilar exceed the potential need for additional cycles to conceive? And do we need to share this data with patient and to discuss whether they will prefer to use the originator or biosimilar this data available? Can you please comment on it?
Thank you. Yeah. One of the key questions I was getting whenever I was sort of presenting the original data, the 2021 systematic review and meta-analysis, many of my colleagues, and rightfully so, would say, yes, but Dr. Venetis, Christos, we understand that there might be a small reduction in live birth rate, and we believe that that's the case because, you've shown it so eloquently. But at the same time, these are more affordable products, and these are what the patients can afford. And that's what actually triggered us to go and do a formal cost effectiveness analysis. And by both the cost effectiveness analysis that I presented, it's quite evident. And obviously, one is done within the Spanish system, the other one is done within the French system. Based on the input, the data, the economic input of those two systems in those two settings, the originator product is the one which is actually cost effective. And it's one of the cases where essentially cheaper is not always better. So I cannot comment on whether that cost effectiveness analysis would apply to other settings. There might be cases where the difference between the biosimilar and the originator is so high where essentially that incremental cost effectiveness ratio is, let's say, many thousand euros, many more than 20, and then the biosimilar becomes the cost-effective product. That's something for essentially its clinician to sort of kind of look at. But certainly based on two different European countries, and there is another one also from Germany, which for the sake of time, I didn't want to present here, you do get more for your buck essentially by using the originator compared to the biosimilar, given this difference in efficacy. So whenever I discuss sort of value for money or cost effectiveness, I always discuss this data with my patients as well. And I say, look, this is not for Greece or for Australia when I was working in Australia, but this very clearly showcases that cheaper is not always better.
Thank you. And one more very concerning question, as I can see it. Are there long-term health data available for children conceived using biosimilars? So what do you think about it?
So far, and I am someone who's very deeply into the long-term health effects and the outcomes of children. So far, there are, to the best of my knowledge, no studies, at least no big data studies that will show that essentially we have exactly the same outcomes in terms of congenital malformation and other long-term outcomes, neurodevelopmental outcomes, overall health, cardiovascular health, and so on. That is not to say that we suspect there would be a difference there. But of course, whenever you start seeing the differences somewhere, you always want to make sure that you stay safe. To the best of my knowledge, such a study explicitly comparing the biosimilars of follitropin alfa with the originator or with other gonadotropins in terms of long-term health outcomes of children has not yet been performed or published.
Okay, thank you. And one more very interesting question. So the most data which came from RCTs is about the first fresh transfer. But we also have some I can say robust data because of the very interesting methodology from the real-world evidence data about the possible difference and the cumulative live birth rate. So it means for me that it might be not about the endometrial receptivity, but about the quality of oocytes and embryos. Could you please comment on the
Yeah, yeah.
possible reasons for
That is a very, very insightful comment. I'm not sure whether it's your comment, Professor Gryshchenko, or one of our the members of the audience, but this is a very insightful comment. These are indirect observations. You're absolutely right. If the effect was solely on the fresh cycle, the cumulative live birth rates would have been potentially, the difference would have been reduced. We do see a small reduction, to be entirely fair, from 0.81 to 0.86, but it's still there. So whether that means that there is absolutely zero effect of the endometrium, or whether that means that there is a small part of the endometrium and another part of essentially the embryo quality, that these are interesting hypotheses, but I'm afraid that especially in the absence of randomized evidence, it is probably too risky to draw any conclusion. Essentially, these are very interesting hypotheses, which again, I would urge anyone who's interested in the topic to try and look into, because we urgently need more data.
Thank you. And going one step back to the ovarian stimulation. So we can see that the duration of ovarian stimulation with biosimilar seems a little bit shorter. So could the shorter stimulation duration with biosimilars negatively affect oocyte and embryo quality, and be the possible reason for these changes? And
So,
could it be adjusted with the dose of dose adjustment might be?
So you're absolutely right. What we saw, and that seems to be supported also with some studies on the glycosylation and the isoforms, we saw that essentially the in vivo biopotency of some biosimilars seem to be stronger. That means that they stimulate harder. That's why, for example, although there was no statistically significant difference, there was that, 0.3 more oocytes with the biosimilars, although not statistically significant. There was that, shorter duration of the follicular phase of the gonadotrophin stimulation by 0.2 days. Even when you looked at the dose of gonadotrophins, slightly lower dose of gonadotrophins, although not significantly so, was seen to be inferred by the data for the biosimilars. That all paints a picture that says probably biosimilars are stimulating slightly harder, more intense. And that might be affecting, for example, and that's why I have a particular interest in endometrial receptivity and progesterone elevation. Maybe there were differences in progesterone just because they are more potent and they stimulate more oocytes, or the same oocytes harder, and the progesterone was elevated, and that is what is leading to the differences in the. But we didn't have any data on progesterone on the day of ACG. So all these, or maybe the shorter follicular phase, or the fact that you get oocytes that have been stimulated for lower days, that means that the quality of these oocytes might be affected. All these are hypotheses, which unfortunately we cannot confirm. So for the second part of your question, could that be adjusted by changing the dose of the biosimilars? Yes, it could. And if that was the reason behind it, then that might be a way for the biosimilars to become equally efficacious by essentially not using the identical dose, by tapering down the dose. But these are hypotheses that unfortunately we cannot, these are interesting hypotheses, but we cannot actually validate them. We don't know whether they are true or not. And I would like to refrain from making assumptions, because whenever we make assumptions, more frequently, than not, we end up in incorrect conclusions. So it's best to form hypotheses and try and do the research and prove that something that we're saying is actually true.
Thank you. And next question. So it seems that after all these studies and meta-analysis and real-world evidence data, it seems that biosimilars have pretty much the same safety profile comparing to originator, which is a little bit surprising for me, because they have a little bit slightly higher bioactivity, and we just like you just told about it. So in a high responders and in PCOS patients, is it safe, and is the safety profile of biosimilars is the same comparing to originator, and can we use it with the approximately same safety in that
Yeah.
type of
So
patients?
thank you. That's once again an interesting question. And again, as I said, we're treading in a very gray area. You're right. OHSS rates were the same, but if you remember, the relative risk was 1.17, essentially hinting to potentially higher OHSS risks for the biosimilars. And that's another point where it shows that potentially biosimilars are more potent, stronger stimulants compared to the originator. And that could be a result of batch-to-batch inconsistencies, or it could be, as we said, the higher in vivo bioactivity due to different glycosylation profiles. So if this is the case, truly, first of all, we don't have any randomized evidence about safety and efficacy in high responders, PCOS patients, or even poor responders. As I said, all the RCTs have been performed in either normal responders, and I think one or two in general population. Or at least undisclosed, and not explicitly normal responders. Therefore, we don't have the randomized evidence to be able to, say with confidence that yes, they're safe or not. But if we look at the writing on the wall that says that they're probably stronger stimulants or stimulators, then one, I mean, you could potentially still use them if you want in PCOS or high responders, but you need to account for the fact that they might be stimulating harder, and therefore either reduce the dose a bit if you feel that that's the right thing to do, or be ready to do a freeze-all. So make sure, for example, you use an antagonist or a PPOS protocol. So if things get out of hand, particularly in terms of OHSS rate, you can still convert to an agonist triggering and a freeze-all and keep your patients safe. But yeah, you're absolutely right. Given what we're observing in normal responders, the safety in high responders is something that also urgently needs to be assessed.
Thank you. And you just answered my next question about the how can IVF clinics minimize risks when choosing between originator and biosimilars. So the very last question, which I want to ask you, can you please share the three possible and most interesting direction in research and comparing biosimilars to originators, or maybe only these biosimilars for the future uh, for our audience?
Thank you. I think, based on the really, really interesting discussion we had, and thank you, Professor Gryshchenko, and the audience for these very, very stimulating questions. I think we already sort of touched on what potentially the three pillars of research should be. One is identifying differences at the molecular level, either with pharmacokinetics and pharmacodynamics, and looking also in things that might seem mundane but might be important, like purity, like batch-to-batch consistency, like how these medications are being transported from the manufacturing factory to the end user, which is the patient. Are there any differences there that might be affecting how this medication actually would act in daily life? That's one. The second one is obviously doing studies, either ideally randomized, but I know a randomized study is not very easy, doing clinical studies and looking at cumulative outcomes, but not just looking at cumulative outcomes, looking holistically at first transfer, cumulatively the number of transfers, but also looking at the number of oocytes, the fertilization rate, and all the embryological outcomes. And these days we have excellent tools. We have, in many cases, time-lapse incubators that can help us have very detailed views of how embryos grow and see whether there are any differences there, any systematic differences there. And the third pillar of research is obviously what we mentioned earlier, looking at long-term outcomes, health outcomes for babies, and of course for mothers as well, because we should never forget that these are medicine, and as medicine being administered for a short period of time, they still might have an effect on the mother. So ensuring that there are no, long-term health outcome differences between mothers having been stimulated with medication A versus medication B should always be a priority. And the same should be obviously for children that have been convinced via these methods, ensuring that we optimize our treatment and our methodology so that we can minimize the risks, if any, to the babies that we help, be conceived and born eventually.
Thank you so much. So evolution is impossible without innovation, but we need to be very careful when deciding how to use the new drugs and new molecules. So, and I would like to sincerely thank you, Professor Venetis, for sharing your expertise and experience. And I also want to thank all of our participants for their engagement and contribution and questions. And we are looking forward to continue research and collaboration to ensure the best outcomes for our patients. Thank you so much.
Thank you, Professor Gryshchenko, and I would like to also thank you for moderating this very, very interesting session. I hope the audience found the presentation at least thought-provoking and stimulating. And yes, you're absolutely right. All these are evidence and data that we should be taking into account whenever we're making everyday decisions for our patients, whenever we counsel our patients, because at the end of the day, all we should care about and all we care about is the best outcomes for our patients. And that's where we should all be investing time and effort. Thank you. And obviously, once again, I would like to thank Sona-Pharm for giving me the opportunity to share these research findings with all my colleagues in Ukraine. Thank you.
Thank you, Professor Venetis.